So my name is Claire Penny, and I'm the 10x Genomics Commercial Lead at Millennium Science. Today's webinar is one I've been genuinely excited about because it sits at the intersection of two things that matter a lot to researchers right now: cutting-edge biology and the very practical challenge of securing funding to pursue it. The goal today is simple. We want to leave you with actionable insights you can apply to your next grant application. Specifically, we'll be exploring how integrating single-cell and spatial biology approaches can meaningfully strengthen your proposal in the eyes of the all-important reviewers. To guide us through that, we have a fantastic lineup of speakers. First up, from 10x, we have Jeff McDermott, Associate Director of Sales of the Sales and Technical Advisory Team here in APAC.
All right. I just want to start off by saying thank you so much for attending the webinar today. My section, I want to talk about how to generate pilot data that really genuinely strengthens your grant applications. And this, to me, isn't just about running bigger experiments or generating more data. It's about designing the right experiments early so that when you're submitting a grant, you're not just presenting a hypothesis, but you're actually presenting evidence. Before we get into the technologies or approaches that I'm going to discuss, I want to take a step back and talk about why pilot data has become so critical in the first place. And it's that strong grants increasingly rely on data, not just hypothesis. And a strong idea still very much is essential. But what I'm hearing more and more from local researchers is that reviewers are expecting evidence that this idea is feasible. From practical perspective, funding is more competitive than ever, budgets are tighter than ever, and there's less room to explore these high-risk ideas. So there's increasing pressure to demonstrate the feasibility earlier on in the process. That's translated into higher expectations from reviewers. And again, I'm hearing from talking with local researchers that they want to see clear evidence that your approach works and that your hypothesis is supported by preliminary data. Importantly, they're not just looking for descriptive data sets, they're looking for biological insight. And often what stands out the most is a clear, compelling figure built from having pilot data. And this is where a well-designed pilot data really becomes very powerful. It allows you to de-risk your hypothesis before committing any significant timing and resources. It gives you early biological insights that can refine or perhaps even reshape your entire approach. It strengthens the overall narrative of your proposal, and importantly, it helps differentiate your application in what is obviously a very competitive space. But the goal isn't just to generate pilot data, it's to generate the right pilot data. So let's explore what I meant by that, about the right data, and we'll use this little example here, which is a human FFPE breast cancer sample. And this is a real-life tissue. It's taken from a patient, and as you can see here, I'm showing the H&E image of this FFPE tissue section. So just like all tissues, this is a complex mixture of cell types and cell states, and of course, they're all communicating and influencing each other. And I guess to further highlight that, I have a small cartoon here of the tumor microenvironment, which shows the complex and dynamic interplay between all the different cells within that spatial niche. Now, traditionally, you may have turned to doing something like bulk RNA sequencing to perform your pilot experiments when using a complex sample like this. So as a researcher, you may want to better understand what is happening in this FFPE tissue. Is there cancer present or is it not present? Is this person responding to a treatment? But of course, the sample is a complex mixture of various cell types. So when you take that bulk measurement, in essence, you're just averaging out the biology. You do get a measurement of all the genes in the transcriptome, but that represents an average signal across the entire tissue. So yes, you've got pilot data, but given the underlying biology of this tissue is extremely complex, the question becomes, is this data actually useful in any meaningful way in advancing your hypothesis, and I guess in overall advancing your grant application? So now I'm going to introduce single-cell RNA sequencing. Again, we're going to ask, what is the expression level of all the genes in the transcriptome? But now we have the power to address this question on a cell-by-cell basis. You're measuring whole transcriptome gene expression from one cell at a time, but you're doing that at a scale of thousands, even right up to millions of single cells. And as you can see, this yields quite literally an explosion of data where you can now deeply profile individual cell types and cell states from complex tissues. And you can see by doing single cell, we now have a significantly deeper understanding of all the cell types present. But cells don't just operate in a vacuum. They're not a single unit. Instead, they interact and influence each other. And that's where spatial biology is so important. And to put it simply, you're connecting the what with the where. Now, not only are you measuring transcripts with single cell resolution, but you also retain all the spatial context of those cells. So if you take that middle plot, now we've placed every single one of those single cells back into the context of the tissue. And from this, you can see which cells are interacting with each other and how they go on to form spatial niches. And hopefully from just this little example here, it's really clear just how important it is to use tools that offer insights at that fundamental resolution of biology, which is the resolution of a single cell when you're generating that high-value pilot data. I really do want you to leave with a clear understanding that not all pilot data is created equal, and that a small number of single cell or spatial experiments can be far more powerful than a large number of bulk measurements in the context of, again, building meaningful pilot data. So here are the tools from 10x Genomics that allow you to provide insights at that fundamental resolution of biology, which again is the resolution of a single cell. And as you can see, we've got 3 platforms that each allow you to take a look at single cell biology with a unique perspective. So there's Chromium single cell, Xenium in situ, and Visium spatial transcriptomics. But rather than focusing, I guess, on the technologies themselves, I want to focus on what they enable in the context of pilot data. So the next few slides are really going to step through what are the pilot scale kits that these technologies have. So when designing pilot experiments, the key question is, what kind of insight do you need to strengthen that grant application? If you're exploring biology and discovering cell types, then single-cell RNA sequencing is typically the starting point for most researchers. For discovery-driven pilot studies, whole transcriptome approaches, such as the universal assays and the flex assays shown here, they allow you to explore biology in an unbiased way. And this is often where you identify the key signals or cell populations that form that basis of your grant hypothesis. So let's look at how the universal assays can help enable pilot studies. You might simply know these as the single-cell 3' and the single-cell 5' assays. They've really been the workhorse of the single-cell community over the past decade, and chances are, if you're looking at single-cell data out the literature, it was generated with using one of these kits here. So as you can see, both assays are based on using a reverse transcription-based biochemistry, where you directly capture and sequence mRNA molecules from single cells. And that's great because they're species agnostic. So you can go beyond human and mouse, and certainly here in the Australia, New Zealand funding environment, people are looking at diverse species, even out to plants. So the core measurement of both these assays here is whole transcriptome gene expression, but you can extend that menu of analytes beyond that. There are cell surface proteins you can measure, and then with the 5' assay, you can also examine full-length paired VDJ sequences from T and B cell surface receptors. So that's perfect if you're an immunologist and you're interested in assaying the immune repertoire. So the single-cell 3' and single-cell 5' assays are extremely valuable tools for generating pilot data. But of course, single-cell has traditionally been more costly assay compared to bulk RNA sequencing. So how can you generate meaningful pilot data without having to invest large amounts of money up front? There's no point in spending your entire grant before you've even actually applied for it. So one of the most practical ways to improve a pilot study design is through sample multiplexing. And that's where, instead of running samples individually, you pull the multiple samples together into a single sample and you run that as a single experiment. And this significantly reduces the cost per sample and allows you to include more conditions or more replicates or whatever you need to strengthen your pilot experiment. So rather than choosing between breadth and budget, you can often achieve both through multiplexing. So in my job, almost everyone I speak with about single cell wants to multiplex samples in some form to save money. And there, as highlighted on this slide, many ways you can do this. But I'll say there's some real practical and technical challenges that you need to consider, particularly in the context of a pilot experiment, which is a bit smaller, maybe your first time doing single cell. So for a pilot, rather than rushing to the most complex forms of multiplexing, it's always my suggestion to first consider a relatively new technology from 10x Genomics that we call on-chip multiplexing. The reason I recommend on-chip multiplexing is that it makes single-cell multiplexing really, really simple and really, really reliable. There are no additional upstream steps, so you can combine to 4 samples per library. And also, this multiplexing doesn't require any extra sequencing or special libraries, so there's no data loss. You're both saving money on every sample, but also every sample is cleanly separated out during the analysis. Other multiplexing approaches, you've got to do sample labeling and much more sample prep here. It's far simpler. So for the sake of time, I'm not going to dive into how this works, but at a high level, you prepare 4 separate samples, you load 4 separate samples onto the microfluidic chip, and then during the single cell workflow, these 4 samples are simply turned into just a single library for sequencing, and then downstream informatics are used to assign every cell back to its given sample. What you can see here in this plot is that we have 8 sample and 16 sample kits. And these are perfectly suited for the number of samples for a small but impactful pilot study. So highly recommend, if you're in that 8 to 16 sample range, really no brainer to use this kit and save a bunch of money to generate pilot data. So moving on to our next assay, which is gene expression flex, and that has strong analytical utility in the context of generating pilot data as well. Just like the previous assay, the core measurement of this technology is whole transcriptome gene expression. Begs the question, why do we have another assay for measuring the same group of analytes? We've got 3 assays for measuring whole transcriptome gene expression. Well, with Flex, the key benefit of this assay and the key reason you want to use it is it allows you to fix and store samples. And this means you can now decouple sample preparation from having to run the actual single cell assay, which is a game changer for a whole number of labs. With Flex, you can take either your suspensions of single cells or actually tissue sections as well, and you simply fix those and then you store them for later use. And this means you can build up a bank of samples that are ready to run in your own time. So this is really great if perhaps you don't have the money for a pilot experiment just yet. You can still do your actual experiment, you still collect those samples, fix those samples, and you store them, and you can run them at a later date. And it's also really well suited for longitudinal pilot studies, or if your samples need to be collected individually. So if you're dealing with patient samples that are coming in, and it's just not practical to run a pilot study every time you get a sample, then you can bank these and then process them all together. Another key advantage of this assay is it also works with FFPE samples. And this is great, again, for pilot studies, because you can access archive samples that already exist. So you may actually want to collect fresh samples for your actual study once that's funded, but with this approach, it allows you to collect data on samples that you already have. So if you've got archive samples and know with relative ease, you can generate single cell data from these FFPE blocks. We recently upgraded the Flex assay, which comes with significant improvements to scale in terms of both cell and sample numbers. And also importantly, it comes with some significant reductions in actual costs. So in certain product configurations, you can profile up to 1 million cells from just a single reaction. And likewise, you can pull anywhere from 2 right up to 384 samples in just a single reaction. But for a pilot experiment, you probably want to be on the other end of the spectrum in terms of cell and sample numbers. You're not rushing into a 1 million cell experiment. And that's why I've highlighted on the plot to the right here that we have a 4 sample kit. And again, this is ideally suited for a small scale pilot. Also, that comes with some significant cost reductions with using that kit as well. But this is a great way to generate some data to show feasibility. And then when you're ready, you can take advantage of the scale abilities of the larger versions of this kit. So far I've focused on single cell approaches and how they can resolve cellular heterogeneity and give you a detailed insight into the cell states. But one key piece that's still missing, and that is spatial context. In most biological systems, it's not just which cells are present that matter, but where they're actually located, how they're organized, and how they interact with each other. And that's where spatial approaches, such as Xenium, really come into it. They allow you to take some same type of molecular information as we just saw, gene expression, but place it back into the tissue, giving you a much more complete picture of the biology. And importantly for pilot data, this often translates into very intuitive and visual outputs that can be incredibly powerful in your grant application. So to showcase the measurement that Xenium is capable of, I'm going to use this FFP human ovarian cancer sample as an example. What you see here is a H&E stain of the tissue, so you retain the ability to perform morphological analyses. But now let's examine the measurement that the Xenium instrument actually makes. And to do this, I'm going to zoom right down to the single cell level of just a random part of the tissue. So Xenium is essentially a highly sophisticated molecular microscope, and it works by taking multiple images of the tissue. So there's no sequencing involved here. First set of images are used for assessing cellular morphology. These are stains added during the sample preparation, which are designed to target cell membranes, intracellular RNA, and nuclei. And the whole purpose of collecting those very pretty looking images is for the instrument to find and segment cells with a high degree of accuracy. Beyond finding and segmenting cells, the instrument will then detect individual RNA molecules directly within the tissue itself, which is what each of the tiny dots on this screen represent. And now there are 2 data layers. The instrument knows where cell boundaries are located, and it knows where individual RNA molecules are also located. So quite simply, all RNA molecules that fall within the same cell boundary are now classified as a single cell. The instrument can then classify these different cell types and cell states, which is what you see here. They're all differently colored. And of course, we're zoomed right into that small section of tissue for this example, but the reality is this signal is acquired across the whole tissue section, which is about 400,000 cells in this case, and about 120 million individual molecules or transcripts. The image you see here isn't a fluorescent image of the tissue, instead it's a molecular image of the tissue, and that's built up from measuring individual RNA molecules. So why is Xenium a great tool for generating pilot data from your tissue sections? So beyond generating transcriptomic data with true single-cell resolutions, as you just saw, there really are two key reasons. The first is the amount of tissue that you can process, and the second is the relative low cost to the platform since there's no sequencing involved, it's just microscopy. So we'll start with examining how much tissue you can fit onto a Xenium slide. The area you have to place tissue is very large. It measures 10.5 by 22.5 millimeters in size, and each Xenium run processes 2 of these slides at a time. It's completely up to you how you fill this area, and all tissue in this area is analyzable. And here's some quick examples of how you can fill the analyzable area. The take home here is this is similar in concept to multiplexing for single cell sequencing. So in there, you're combining multiple samples into one sample to run per reaction. Here, you're multiplexing through placing multiple tissue sections onto that Xenium slide. So to summarize, pilot data has really become essential for competitive grant applications, but it's not about generating more data, it's about generating the right data that clearly supports your hypothesis. Single cell and spatial approaches allow you to maximize the insight you get from relatively small amount of experiments. And of course, multiplexing helps you do that in a cost-effective way, whether that's multiplexing for single cell by combining multiple samples into one, or multiplexing for spatial where you're putting multiple tissues on that slide. Ultimately, the biggest impact comes from thoughtful experimental design, making sure you're generating data that directly strengthens your grant application. And then just want to finish on this slide here, which is supporting your project from design to discovery. It's not just about buying reagents from 10x and generating your own pilot data. We also have other resources to help you with your grant application process. So we have grant application resources. These are small documents written to give you overviews of the assay, what they're capable of, and help you sort of take paragraphs from here and put it into your grant. They're hosted on our 10x website as well as the Millennium website. Obviously, spending money to generate data can be tricky for some groups. So we also have freely available data sets. You might not have the exact data set that matches, the biology you want to measure in your grants, but at least you've got something. So you can download these for free. You can see we have them across all of our platforms. Also, not just 10x hosts these kind of data sets, but there are a number of websites as well that have large repositories of existing data where you might not need to actually go and reinvent the wheel. And finally, technical consultation. That's what I do at 10x. That's what I spend a lot of my time doing throughout the day, is meeting with groups in a sort of a smaller setting, perhaps even one-on-one, and talking through your project. What is it you want to do? What is the key biological measurement you want to make? How many samples do you have? And then working backwards from that goal, connecting it with the products we've got and how to do it all in the most efficient way possible. So with that, again, I just want to say thanks for chiming in today, and I'm going to hand it across to the other speakers.
So now we'll hear from Millennium's own marketing coordinator, Lim Chi Lu, who joined us from a postdoc position at La Trobe. So she brings a really unique perspective, bridging the research and industry sides of this conversation. Thanks, Lim Chi.
Thank you. Can you hear me and see my slide okay? Yep, okay. All right, yeah, thank you, Claire, and thank you, Jeff, for the introduction. So Jeff basically highlight all the points that I want to go through for my grant application. So here I will be just showcase how I practically put that in my grant applications. So I'm Lim Chi, I'm a researcher from La Trobe University. And together with University of Adelaide and Southern Cross University, we received about 1 million grant over 4 years from the recent ARC Discovery projects. So the projects have one fundamental question to tackle, is how do we grow more food with less water? And these projects sit in the intersection of food security and climate resilience, two of the Australia's most pressing challenges. So the team bring together some experts in plant physiology, biophysics, and some cutting-edge technology to answer this question. So my part in the team will be the single-cell transcriptomics. So let's start with some background of the project. So crop water use efficiency is linked directly to the crop yield. and it's very limited by our ability to understand how plant cells control water loss and how they respond to stress. And gut cells, which show in the figure here, are the gatekeeper for the transpiration, the plant water loss, as they control the opening and closing of the stomata. Yet they represent a very small and transcriptionally distinct population that embedded within the whole complex leaf tissue. So historically, we rely on bulk assays such as bulk RNA sequencing that average the signal across many different cell types. So these can obscure the very regulator that determine the stomata and the gut cell behavior. So to make meaningful progress on the water use efficiency study, we need some platform or some infrastructure that can allow us to see the regulation at the level that where it happened, that is the cell level. So the overall project aim is very precise, is to identify and validate the novel molecular control that reduce water loss, but without compromising the crop yield. So it's a balance that proven to be very difficult using the traditional conventional approaches. So the innovation of this grant proposal is not simply include all the different advanced techniques and technology, is the way that how they're being architecture around the cell type resolutions. So autogenetics allow us to disturb the signaling with temporal precisions, and phosphoproteomics capture the rapid downstream events happening, and single cell sequencing here will allow us to identify the spatial and cellular context required to interpret all those signals correctly. So without single cell, we won't be able to assign or prioritize the target with confidence. So in this project, the single cell sequencing function as a integrating framework, try to make the rest of the pipeline more interpretable and actionable. And then from there, we can do some functional validation using synthetic biology. So why bulk approach are no longer sufficient? Why single cell resolution is essential? So Jeff touched on that briefly. So this is the question that reviewer will be asking or thinking in their mind as well. So bulk RNA sequencing average the signal across different cell type. So in doing so, it make the very response that we're trying to capture hard to be captured. So, and then the second point is gut cell, the cell controlling the opening and closing of the stomata. Eventually, the water loss is a very small population embedded in the sea of other leaf tissues, leaf cells. So their signal will be very diluted, distorted, and effectively invisible if you are using the bulk RNA sequencing approach. So single cell so what we try to highlight in this grant application is the single cell resolution isn't a nice to have, but it's the only way to answer the biological question here. So that requirement fundamentally shift the single cell sequencing being a nice to have in this grant application to a core experimental platform to have. And we didn't just come to this hypothesis alone in the grant, we came with some data to support our grant applications. So the first step we did is to optimize the nuclei isolation on the leaf, which are our target tissue, using a transgenic line expressing a gut cell specific reporter gene. So as you can see in the microscopy photo here, we use the transgenic line to optimize the nuclei isolation to make sure we can successfully isolate nuclei from the gut cell, which is standard in yellow here. And on the nuclei isolation, we can clearly see we managed to capture some nuclei that have yellow fluorescence. So after successfully optimize the nuclei isolation, we perform single nuclei RNA sequencing, and then we conducted a clustering analysis and use some well-established cell type marker from the literature to annotate those clusters. So this approach allows us to generate pilot single-cell data specifically to address reviewer concern about the feasibility and the biological resolutions. So based on this data, we can clearly demonstrate that there's a clear separation of gut cell from the rest of the leaf cell type, including mesophyll cell, vasculature cell, and epidermis populations. So what we want the reviewer to see is single-cell sequencing is not an expensive add-on to the project, but a fundamentally discovery platform. So we prove that we have the mature workflow and we solve the hard problem, which is extracting the nuclei from the plant leaf tissue. And we also show that our experimental design is scalable using multiplexing tool provided by the platform. So this platform not only generate discovery across the life of the project and beyond the project as well. So we really highlight that in the grant application that to the reviewer that the proof of concept of the workflow is robust and biological meaningful, and the platform is proven, is not speculative. So in the funder project, the single-cell sequencing also underpin the discovery of the entire project. So by integrating with the phosphoproteomics, we aim to map rapid signaling event that happen to the transcriptional outcome in a specific cell type that is gut cell, rather than inferring the response from the mixed population. So this will enable us to resolve gut cell specific response alongside with broader leaf signaling to identify novel regulator of the stomata dynamics and then prioritize the candidate for functional validation. So importantly, this approach reduce the false positive and also increase the translation by ensuring that the target are relevant at the cellular context before moving to validation using CRISPR-Cas9 knockout or overexpression study, and ultimately translate that to other crop species like canola. So a key concern for any applied biological grant is off-target effect. So what we highlight in the grant application is by working at the cell type resolution, we can identify the target that are specifically to gut cell, which means the intervention that we develop are precise and reduce the list of pleiotropic effect at the broader plant level. So this de-risks the translation pathway to canola and other crops, and it directly strengthens the impact of this application and highlight that the single cell data directly drive the experimental decision, and the downstream investment is not just a data sets. So finally, I want to emphasize that what we're building on the grant application is not just a platform. The workflow we develop will be reproducible and scalable through multiplexing and other future funding opportunity and collaboration programs, including translating it to other crop species. So the reviewer should see this as a competitive advantage, the capability that position this project and Australian plant science at an international frontier. So thank you. I hope my example demonstrate that the technology is no longer a new and emerging technology, but a mature one that you can include it in your grant to demonstrate that kind of platform is it's not fashionable, but it's the only way to answer certain biological question with precision and robustness that grant nowadays the competitive grants demand. Yep, thank you.
Thank you so much, Lim Chi. Okay, so now we'll hear from Professor Grant Drummond, Professor of Physiology and Co-Director of the Centre for Cardiovascular Biology and Disease Research at La Trobe University, alongside postdoctoral researcher Erica Figueiredo Galvao. They'll be sharing their first-hand experience of how pilot data played a role in their grant success. So thank you so much. Looking forward to this.
Thanks, Claire. I think I'm gonna set the scene for this presentation. Erica's controlling the slide, so I might just get Erica to flick through to the next slide, if that's okay. So again, just to set the scene for the person that you really want to hear from, that being Erica. My name's Grant Drummond. I co-direct the Centre for Cardiovascular Biology and Disease Research at La Trobe University. Our centre has around about 60 staff and students working mainly across sort of basic or preclinical cardiovascular research. So I guess one of the major challenges that creates for us, and I'm sure this resonates with many of you, is that many of the sort of the medically related granting schemes to which we apply, NHMRC, MRFF and the Heart Foundation, not only are they becoming increasingly competitive, but there's a much stronger emphasis on sort of significance, feasibility, and in particular, translation. So for those of us working primarily in animal models, that can create a real challenge. As you can appreciate, access to human tissues or prospective clinical samples is often limited, slow, or even just not feasible at the sort of the early stages of a project when you're putting in your grant application. And of course, you know, we all know that one of the main things that assessors are now asking of us are, how do you know that this preliminary data or this idea is relevant to human disease? So what our approach to kind of addressing that question is to access or leverage publicly available human data sets. Originally, we were kind of focused on sort of hospital data records, but over the past couple of years, we've had more and more focus on looking at sort of these publicly available single-cell transcriptomic data sets to directly test whether the pathways and mechanisms that we're identifying in our animal models are also evident in humans. So from an NHMRC perspective, this has been really powerful for our group. It's allowed us to really de-risk our applications. It strengthens the significance and translation sections of the grant. We're using it to provide human relevant preliminary data. And it also gives assessors confidence that the biology we're pursuing isn't just sort of technically sound, which we can control, but that it's sort of genuinely disease relevant. I guess with that, I want to be really clear. I don't actually have the data science expertise to do this myself. The reason this approach has worked for our group is because we're fortunate enough to have someone like Erica working with us. Erica's been able to sort of interrogate these data sets rigorously and appropriately. And that combination, the strong mechanistic biology that we bring with the high quality human data analysis, has become a really effective way we've found for us to improve our feasibility, reduce our risk, and ultimately write more competitive in NHMRC grants. So with that, I'm going to hand over to Erica, who will give you some sort of practical examples of the way we've been approaching this.
Thanks so much for that, Grant. And so today I'm going to give you 3 examples, 2 of which that have led to successful NHMRC ideas grants, and one of which we're currently working on. And well, let's just get into it. So for the first one, we put in an application looking at the cognitive impairment in postmenopausal hypertension. And you may or may not be aware of this, but menopause is actually associated with a 2-fold increase in the risk of hypertension. And postmenopausal women are also at a higher risk of developing cognitive impairments. And we also had a little bit of preliminary data from one of our collaborators who ended up being a chief investigator on this grant that showed that hypertension actually exacerbated that cognitive decline in postmenopausal women, so much so that you could actually see brain shrinkage before the cognitive impairments were even there. And we got to thinking about, well, if this is the case, why didn't hormone replacement therapy work in the past? And because there have been a couple of human clinical trials out there that actually showed that HRT or hormone replacement therapy actually increases the risk of cardiovascular diseases, strokes, and dementia. And we hypothesized that this was due to 2 types of estrogen receptors. So The one that most people are familiar with are the nuclear receptors, the estrogen receptor alpha and beta, but there's also a novel receptor that's on the cell surface of cells, which is called the G protein-coupled estrogen receptor. Now, we had some initial bulk RNA sequencing data that showed that this cell surface receptor, the G protein-coupled estrogen receptor, was reduced in hypertension. But as Jeff quite eloquently pointed out before, bulk RNA doesn't really give us that insight into which cell types are actually expressing the genes of interest. So we wondered, where are those receptors actually being expressed, and is their expression comparable between mice and humans? So for this, we included data from these two single-cell transcriptomic data sets, and I'm hoping you can appreciate here that we started off by just looking at which cells were expressing these genes, and we can see that the nuclear receptor was only expressed in the neurons in both mice and humans, but that the cell surface receptor was actually primarily expressed within endothelial cells. So this is one of those few examples where the data is actually really quite clear cut, but we saw this and we thought, wow, this actually de-risks our grant a lot, because it shows us exactly where those receptors are being expressed. And with this data, we were able to put together our hypothesis. So for this grant, we said, well, we think the reason that menopause is associated with an increased risk in cognitive decline, and why hormone replacement therapy doesn't work, is because in premenopausal women and men, you've got this nice balance between the cell surface G protein coupled receptors and the nuclear receptors. So when either endogenous estrogen or hormone replacement therapy activates these receptors, you get an increase in blood perfusion and an increase in neuronal activity. But in hypertension, like I said before, we're getting that reduction in this G protein coupled receptor, which, thanks to that single cell data, we know is now a reduction in the expression in the endothelial cells. And we hypothesized that in this case, when you give someone HRT, you are increasing that neuronal activity, but you're not increasing the blood flow. So this leads to a mismatch in supply and demand, where you're getting an increase in cognitive activation, but you don't have the appropriate blood supply to maintain that. And we had a couple of mouse models in there that also backed some of this up, and we actually ended up securing about 1.5 million in NHMIC Ideas funding from this project. Now, the second project that we worked on was actually on interleukin-18, which is a pro-inflammatory cytokine, and its involvement in abdominal aortic aneurysms, which is a life-threatening condition that we currently have no pharmacological treatment options for. The only option available for patients is surgery, and even then the surgery is so risky that doctors don't usually turn to it unless the benefits outweigh the risk of the surgery itself. So we thought this would be an interesting, I guess, end organ damage or disease to study. And we had a really striking animal model, where we saw 0 incidence of abdominal aortic aneurysms when we genetically depleted interleukin-18. But what we wanted to know, and what we felt was missing from our data, was the mechanism. So we wanted to know which cells secreted this interleukin-18 and which cells responded to it, so through the receptor expression. And again, we wanted to see if this data was comparable between mice and humans. Now, here I'm really only showing the human data set, but we also had our own mouse data set for this, which pretty much highlighted the same things I'm showing you here. So in the healthy tissue, you've got very little immune cells in there, but in the disease tissue, you get this massive infiltration of these immune cells. And that was great, because our mouse data reflected the human condition. And then we thought, well, does the mechanism also reflect that? And what we can see in these plots is that the myeloid cells, like monocytes and macrophages, are the ones that were primarily producing this pro-inflammatory cytokine, and that these immune cells, like T cells, NK cells, were the ones that had the receptors for it. So we knew it had something to do with these immune cells. And this allowed us to build a very mechanistic NHMRC ideas grant as well, where we could target the interleukin-18 pathways at different time points, but we could also target those immune cells like the T cells and the natural killer cells a little bit more specifically. Now, we are also putting this project forward soon, and we don't know what the outcome for that is yet. But for this one, we wanted to dig a little bit deeper and go beyond just gene expression and where they're being expressed. So I have been working for the past couple of years on B cells and what their role is in hypertensive diseases like chronic kidney disease. And the very first question we had was actually which of these B cells is involved in this response, because there are a lot of them. And then we wanted to figure out which mechanism that they're doing this by, and whether this, again, is comparable between mice and humans. And from my work, which is available as a preprint, but we're submitting for peer review soon, we found that one of these B cell populations was primarily involved in these hypertensive diseases, and that was our marginal zone B cells, which I'm hoping you can appreciate increase in hypertensive animals. But one of the things I often get asked whenever I present this data is, well, what happens in humans? So I went looking to see if I could find any kind of single cell data set out there that had samples from hypertensive patients. And I got quite lucky in finding this publication just last year, a year and a bit ago. And they looked at people with chronic kidney disease, and they had a mixture of diabetic chronic kidney disease and hypertensive chronic kidney disease. So I went in and started analyzing that data, and I focused primarily on the patients that had kidney disease due to hypertension. And honestly, it was quite striking. The similarities. So in the chronic kidney disease, we're seeing more than 10-fold increase in these memory B cells, which have a marginal zone B cell phenotype. So this really highlighted to us that this increase in marginal zone B cells is not just in mice, it's actually in humans as well. And then we turn to ligand receptor analysis, which is another really powerful tool that is available from these single cell data sets, because you can look at which cells are expressing a ligand and which cells are expressing the receptor for that ligand, and calculate the probability that those populations are actually communicating with each other. And for both the mouse shown here in the top and the human in the bottom, we saw an increase in antigen presentation specifically in the marginal zone B cells. So it looks like these cells are presenting something that's triggering the immune system, that's driving this hypertensive disease. And this is evident in both mice and the humans, although in the mice it's a little bit more obvious than the humans, but we're not too worried about that. The other key thing we found is that these marginal zones are the initiators. So you can see here they're increasing their outgoing communication. And the main receivers are the CD8 T cells, which there's a vast amount of literature out there showing that these cells are effector cells and that they have a prominent role. So we think that these two cells are working together and promoting disease. So we then hypothesized that these cells are critical mediators of hypertension because they amplify that immune response and get other immune cells to target specific things in these tissues. And a big part of what we want to do next is identify what that is. Now, I've done a little bit of work trying to get various different preliminary data for a couple of different grants now, as you can see. And I want to echo Jeff's point earlier that not all data sets are equal. Some of these you have to take into account where you're going to get it from. I find that the best data set is often your own, because we work in very, very specific fields. And that often means that the data that you're after is probably not exactly already out there, but you can still look for data sets like we did that just helped us illustrate the point that we were trying to get across. And they're not always the perfect data set, but you work with what you got. And so you can, I've found most helpful the 10x databases, and also these public repositories like the GEO, EMBO, and the DNA database. So these are maintained by the US, Europe, and Japan, for example. There are also some bioinformatics packages out there that have data that's already completely processed. So if you're not as familiar with the data set, you can use it as well. But these are often not single cell, they're just bulk or microRNA data from cell-sorted cultures, which is still helpful, but not as good. The other thing to take into consideration before doing this is the state of the data. So a lot of the publicly available data sets are raw or not fully annotated, so you have to know the quality control steps to take into account before you actually get your figures together for these applications. Sometimes they're not annotated, sometimes they are. And if they're not, then you need to allow extra time to do that, especially if you're not familiar with the kind of cells and the genes that they express in a particular tissue. And you also need to consider how deep you want your analysis to go. So like for projects 1 and 2, it was pretty straightforward. We were more interested in which genes were expressed in which cells and whether that fit with our hypothesis. But for our third project, we're going a little bit more in depth because we want that mechanistic insight. So we then have to do downstream analyses. But I think these examples highlight just the versatility and how not comparable, but how complementary these approaches are. And they really, like Grant said, de-risked our applications because they highlighted that actually a lot of these processes exist in both. So the likelihood that this will be able to be translated into humans is a little bit better. And with that, I'll just acknowledge our center.
Fantastic. Thank you. I'll just quickly share my screen here. Okay, so as Erica highlighted so beautifully, publicly available datasets are a fantastic resource. But should you want to generate your own data, I have highlighted here the huge number of local core facilities that can work with you to run pilots. Here at Millennium Science, we also have a number of demo instruments that can be placed in your lab at no cost in order to facilitate you getting hands-on experience of the workflow and to allow you to run. So please don't consider instrument access a limiting factor to incorporating 10x genomics into your application. And I will just note, we're very keen to support pilot projects. We have a number of streams that can help break down the cost barrier for pilot data generation. So do reach out after this webinar if you'd like to talk about this further.